Recording. All right, let's begin. Are there any questions before we begin? I'm sharing my screen. We don't need that. I'm sure there's nothing to see at the moment, but I'm sharing my screen. So today is October 5th. Let me remind you that, oops, let me go up a little bit. If you have not taken the practice quiz 00, you still can until 11:59 p.m. tonight. I would appreciate it if you did try, because if you get it up and running, then you've tested it and you have it on your system. If you have trouble getting it running on your system, you'll need to contact the computer help desk, and they will help you change a setting or two, I don't know, on your computer to get it running. Today, we're going to be talking about Lab 2, aseptic transfers. And let me remind you, you should read the textbook through chapter 3 on the section on compound light microscopy before we begin the labs next week, labs 3 and 4. You should also know that we began the infectious disease project, so you should be thinking about what project you would like to do. It needs to be an infectious disease, and you need to get it approved by me before you begin work on the project. I only want one student to work on each project, so everyone's going to have a different project. And I forgot to mention, the main reason for doing this is to make you an expert on one infectious disease. And it's not, I don't know, going to help you too much in your career, but it will get you started, assuming you work in a career that works with infectious diseases. So it's just a start. So you need to clear it with me beforehand. Sometimes I can break the disease into different categories, like influenza can be broken down into influenza A, influenza B, influenza C. And then if need be, I can divide AIDS into HIV-1 causing AIDS and HIV-2 causing AIDS. So I can break down some projects if there's much desire. So far, I think there's only been 2 people asking for tuberculosis, but I think it was both the first and the second person asking for that one. So don't bother asking for tuberculosis. That one has already been taken, and I don't have a good way to split that one. I can't think of a good way to split that one. Yeah. All right. So let's begin lab 2 if there aren't any questions. If there's any questions, please ask. All right. Let's begin lab 2. So lab module 2, aseptic transfers. The learning objectives are: 1, understand the purpose of using aseptic transfer techniques. 2, determine if aseptic transfer was used from the data provided, meaning we'll give you a picture and then you tell us if aseptic transfer was used. And then 3, define the following terms. What's ubiquitous mean? What's aseptic transfer? What's inoculation? And what's a contaminant? Any question about the objectives? So in our local environment, microorganisms are ubiquitous, meaning they are essentially everywhere. This is a problem when working in the microbiology lab, because generally we want to work with a pure culture, a culture containing one and only one species of organism. Louis Pasteur developed the technique of making aseptic transfers to minimize the risk of culture contamination. And I did also mention that he came up with a technique for getting a pure culture. And what he did was he used a liquid culture and had his organism of interest, which would probably be a mixed culture because he hadn't gotten it pure yet. And then what he did was he made a bunch of dilutions to his culture of interest. And then he replicated each dilution, meaning each dilution had 10 tubes that he inoculated with the bacteria in that dilution. In any dilution that there was only one of the 10 tubes that had growth in it, he reasoned that that was likely growth from one or very few cells. And so it was likely to be a pure culture. And he was correct. It was likely to be a pure culture. It's not 100% guarantee it's pure, but it was likely to be a pure culture. And that was the first way we had to get pure cultures. Of course, Robert Koch came with an easier way to get a pure culture, and you just streak out the bacteria on a solid surface like a petri dish with agar, and then grow it up. And as long as you get isolated colonies, if you take the cells from one isolated colony and then streak it out on a second plate, meaning you take the cells and you get go through two colony isolation events, those cells are more than 95% likely to be a pure culture. But you do need to streak it out 2 times before you can say that it is a pure culture. If you only streak it out 1 time, the chance of it being a pure culture is only about 70%. And that's not good enough in microbiology. All right, we're going to learn how to make aseptic transfers in this lab. During inoculation, something you want to do while you're doing aseptic transfers is to never lay the test tube or the petri dish lids on the workbench, because if you do, they can pick up bacteria from the workbench. And of course, you never lay the loop down once you have it sterilized, because if you put it down, it can touch the surface and then pick up bacteria. To minimize contamination when you're inoculating media, you want to tilt a tube at an angle such as this. You don't want to put it in, I don't know if you can see this. Maybe I put it in front of my face, you can see it. You don't want to put it in this way because any microbe in the air can fall down and enter the tube. You want to hold it at an angle and inoculate it that way. So that way, most microbes falling down from the air are going to hit the side of the tube or miss the opening. There will be some coming directly here that can contaminate the tube. So there is a risk that the tube could be contaminated simply by opening it. Okay, but if you hold it at an angle, there's much less risk than if you hold it straight up and down. Any question about that? Another thing is when you're doing a tube, and this will be really difficult for me to do because I don't have a tube here. Maybe that'll work. I'm going to grab something, just a minute. Yeah, this looks very much like a tube. It's not, but it looks like a tube. What you want to do when you're working with the tube is you don't put this down on the workbench. And I know you can't see that. If I go like that, you can probably see it. But I'm not sure because it's not in front of me, so maybe you can't see it. What you want to do is hold the tube at an angle and then pretend the pencil is a loop. You want to hold the cap in your hand like this. Hopefully you guys can see this. Can you guys see this? Sort of as the picture is showing.
Yeah, a little bit.
Is it just because it's not showing up or what?
No, that's better when you hold it up a little higher.
Okay, to my face, it usually works. All right. And the other thing you can do while you're doing a plate, when you're streaking a plate, is number one, keep the plate uncovered for as little time as you can, meaning do your work quickly and then cover it. And second, while you're doing your plate, I don't know if you can see this, but let me put it in here. This will be hard for me to do because I gotta hold so many things here. You want to hold the lid partially over the plate and put your loop inside and now streak it. And the best thing is look at the picture here. So that if something comes down from the air, it's going to fall down on the top of the lid, as opposed to there will be a risk that it can fall right here and get into the auger. But if it falls here, it won't get into the auger. So you want to cover the plate as much as you can while you're working and then leave it open for as little time as you can. Any question about any of that? Okay, it goes without saying. Do not talk or sing or anything like that while you're transferring, inoculating a tube or a plate. And if you feel like you're going to sneeze, you should cover the plate before or the tube before you sneeze or before you cough. And obviously, if you do sneeze on your plate, you should probably just throw it away and go get another plate. You should also sterilize the inoculating loop or needle before you work. Sterilize the forceps, which we do use a forceps in this lab. Disinfect the workstation, meaning the area you're working with, sterilize it or disinfectant before you begin work. And then it helps if you're wearing a lab coat and gloves and washing your hands. In order to inoculate, that's transfer new sterile media, we must make sure that we sterilize everything that can potentially contaminate the sterile media. How you do that is you only use one sterile media, and two, you use something like a loop here, and then you sterilize the wire portion of the loop, this region here. You do not sterilize the metal portion or the plastic or rubber hand portion. If you do that, it'll become too hot to hold. You only sterilize the loop. And how you do that is you put it in either the flame, which we almost never use the flame in the microbiology lab anymore. We have bacto-incinerators, which you turn it on and about 5 minutes later, the stone inside the bacto-incinerator will be red hot, and you put the loop inside the bacto-incinerator until the loop becomes red hot, and you can actually see it. And once it's red hot, you bring it out, and you then cool it. We'll talk about how you do that, and then use it to pick up your cells and inoculate the new sterile media. To know the terms of this lab, in this lab, you're not going to be physically performing any of these activities because all of this lab is going to be online. But you will be watching videos of these activities, and we expect that you will know and understand the material in these videos and in this lab. In procedure one, you're going to be transferring sterile liquid water using a glass pipette into a tube of media. So procedure one is through here. You can go ahead and read it. You should obviously appropriately label all your tubes and your plates with more than just your initials and the date. It's not stated, but you should have the media in there, what you're inoculating, and then the temperature is optional. Anyways, when you're using a pipette or a burette to measure water, you will notice that the water will not go across flatly. It'll form a meniscus like this, meaning it'll bend at the sides. And how you read the meniscus is you read from the bottom of the meniscus. You do not read from the middle of the meniscus. You do not read from the top of the meniscus. You then transfer 1 ml of sterile water in procedure 1 into 9 ml of media. I'm going to talk about each procedure separately. This is what the media, the tube of media will look like before you put the water in it, okay? And you should note whether that's clear or cloudy. And then after procedure 1, use this table to interpret the results, or not interpret the results, read the results and interpret the conclusion. Remember, the results are your observation. The conclusions are your interpretations of the observation. So if the nutrient broth is clear for procedure 1, then the conclusion is no organisms are growing in the broth and aseptic transfer was successful. Because remember, we're transferring sterile water into the broth. So we expect nothing to grow in the tube. If, on the other hand, the nutrient broth is turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer was unsuccessful. There will be questions in the lab asking you to write up the result and the conclusions, and your wording has to be similar to the wording in the table. It doesn't have to be the same, but for example, in your conclusion, you have to talk about whether organisms are growing or not for procedure 1 in the broth, and also whether aseptic transfer was successful or not. Any question about any of that? All right, let's go down to procedure 1. This picture is the picture of procedure 1. And this is telling you what was added for each of the procedures. You'll have it labeled question 0. So this is a question, and I just labeled it 0 in your copy. I think I don't have it. It's not in my copy that I do for the teacher's version, but I think it is 0 in the full version, which I don't have highlighted, so I haven't, I don't use this to talk about. Nope, I don't have it in my version, but hopefully you have it in your version, where I'll have question 0. Describe the initial clarity of the nutrient broth prior to inoculating, prior to adding the sterile water. And that's for procedure 1. In procedure 2, you're going to add a paper disc. Is it clear or turbid? And then in procedure 1, answer question 1. Describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation at 35 degrees. Was it clear or turbid? Based on these results, what conclusion can be drawn? So give the conclusion here, and then that's P1A, P1B, list the conclusion here. P13 lists 3 sources of error with aseptic transfer that could explain why the nutrient broth showed growth of microbes. So give 3 possible errors. Any question about procedure 1? All right, procedure 2, you're going to be transferring a sterile paper disc using sterile forceps. And we do have a video for you to watch demonstrating proper aseptic technique. So you should watch that video while doing this lab. In procedure 2, we have the technique here. Use aseptic technique, transferring a sterile paper disc into the nutrient broth. So the paper disc is sterile, the nutrient broth is sterile, and the forceps are well, they're clean, they're disinfected, they're not exactly sterile. We don't make the forceps red hot, otherwise you wouldn't be able to handle it. How you clean forceps or disinfect forceps is you put them in rubbing alcohol, and then you set the rubbing alcohol on fire to sterilize or clean the forceps. And hopefully you'll see a video of that. If not, I just told you about it. So use the results for procedure 2 in this table. It's the same results as for procedure 2. And then the conclusions will be the same too. Any question about that? All right, there's the tube for procedure 2. You can actually see the paper disc at the bottom of it. And then answer question 2 for procedure 2. Describe the clarity of the nutrient broth after inoculating with a sterile paper disc and overnight incubation. Was it clear or turbid? And that's question 2A. For 2B, based on these results, what conclusion can be drawn? And remember, your conclusion should be similar to the conclusion in the table. For procedure 3, we're going to be transferring Serratia marcescens into a tube of broth. For procedures 3 and 4, we have a little video for you to watch to correctly transfer from a sterile, not a sterile, a bacterial stock culture into a sterile broth tube. And there's a little video right there. Some important notes about this second video. You only need to watch until 4 minutes and 23 seconds into the video. After that, you can stop the video. The bacterium being transferred in the video is E. coli, and we're going to use Serratia marcescens, which is a pigmented bacteria. So E. coli is non-pigmented, Serratia marcescens is pigmented, and that will allow you to answer the questions a little more easily. And then you should note there's an error made when the student puts the loop into the E. coli stock tube. It's between 3 minutes and 5 seconds and 3 minutes and 2 seconds. You should try and find what that arrow is. And hint, you can hear some sizzling when he does the arrow. Okay? So for procedure 3, we're adding Serratia marcescens to a sterile broth tube. And Serratia marcescens will be pinkish when it is grown in a broth tube. Broth cultures are problematic because it's difficult to determine if contamination has occurred because all the cells are going to mix together in a broth. You would be able to see if there was major contamination in the tube, meaning if more than 50% of the cells were the contaminant, the color of the tube would look inappropriate. However, if there's slight contamination in the tube, there's no way to know in a broth culture because the cells are all going to mix together. And if there's slight contamination, the tube will still appear pinkish, like that. If you get slight contamination growing in the tube, and the major growth in the tube is the organism of interest. All right, so that's procedure 3. You can read about the procedure. And then for procedure 3, you use the results in this table here. So if the nutrient broth is clear, the conclusion is no organisms are growing in the broth and aseptic transfer was unsuccessful. If, on the other hand, nutrient broth is turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer was successful. For procedure 3, you have the tube here. and then answer the questions. For procedure 3, at question 3, describe the clarity of the nutrient broth after inoculating with Serratia marcescens and overnight incubation at 30 degrees Celsius. Was the tube clear or turbid? 3B, based on these results, what conclusion can be drawn? 3C, how do you know whether you transferred the correct microbe, Serratia marcescens? And as I stated, you should be able to know by the color of the tube or the pigmentation of the tube. All right, for procedure 4.
Can you just go back to that table real quick with the results? I just want to compare mine.
It should be the same.
Okay. I thought it said otherwise, but we're good. Thank you.
All right. In procedure 4, you're going to be transferring, using a loop, Serratia marcescens with sterile technique from the stock culture onto an auger slant. Here is an auger slant here, and how you inoculate the auger slant is you sterilize the loop, cool it on the inside of the tube of the stock culture, and then pick up the bacteria on the stock culture, and then inoculate the slant using a S-like motion. Contamination will be easier to see on a slant because if anywhere on the streak it is not a reddish color, which is the color of Serratia marcescens when grown on a slant, then you know you have a contaminant. Like if this region right here was white or off-white or any color other than reddish, then you know you have a contaminant right there. And that would be a slight contamination because the majority of the tube is inoculated with the culture of interest. But you can tell because the bacteria do not mix together on a slant. Interpretation for procedure 4 is in this table here. If the slant does not show confluent or colony growth, then no organisms are growing on the slant and aseptic transfer was unsuccessful. If, on the other hand, the slant does show growth, confluent or colony growth on the slant, then organisms are growing on the slant and aseptic transfer was successful. There's the tube of the slant after we grew it in the incubator for a day at 30 degrees C. And then answer the questions for procedure 4, which are in question 4. 4A, is there bacterial growth on the slant? And just answer yes or no. And how do you know? Give me a discussion for why you know there's bacteria growing on the slant. And 4C, does it appear to be the bacterium you inoculated or a contaminant? And then that's it for the lab. Any question about any of that? All right, remember to only fill out the worksheet, which I never showed you. So put your answers on the worksheet here. And then submit the worksheet. Only submit the worksheet. Do not submit the lab module. The lab module is much greater in size than the worksheet. And if everybody submitted the lab module, my Canvas website would soon become full, and then you guys would be unable to submit your assignments. So don't fill up my Canvas website. All right, any questions about any of this? If not, I will be here until 8 o'clock or the last student leaves, which will probably be much sooner than 8 o'clock. And I'll be here to answer any questions or help you with the lab. But I will log out once the last student logs off. Any questions? All right, I'm going to stop the recording.